situated here. Okay, so good morning, everybody. I enjoyed my conversations, as I said yesterday, with everybody so far, and I look forward to the small chunk I have this afternoon. Well, this afternoon for me, morning for you all. So today I'm giving my vision talk on some of the work that I want to do in my independent career as a faculty member. If I can get my laser pointer to work, there we go. And really looking at trying to harness these functions of these living polymeric material systems. In case some of you missed my talk yesterday, I'll give you a little bit of overview of my research background. So I got my PhD with Al Crosby, where I did uniaxial tensile testing of ultra-thin films, as many of you heard, where I did instrumentation development, where tensile testers for either liquid supported or freestanding films, and use these instruments to do structure property relationships for our films down to 10 nanometers in thickness, and looked at how the molecular changes in those films related to the mechanical properties. While yesterday I mostly focused on polystyrene, I also did work on polymethyl methacrylate silicon nanocomposites, which is a collaboration with Brno University of Technology, where one of their grad students came in and we measured their materials. And the other project that I worked on a different system was polycarbonate, where we had a visiting researcher from the Korea Research Institute of Chemical Technology who came in to learn our instrumentation and then was working with polycarbonate with me. And so we were looking at the thickness effects and if we could increase their strength through solvent annealing, which you can, is what we found. So that's a very interesting paper if you're interested, or I'm welcome to talk later. So now at Princeton, I'm working with Sujit Datta on the structure property relationships of engineered living materials, where I'm doing spatial programming of biofilms, these bacteria that form their own polymeric substance, which you can control through genetic engineering. So we're doing knockouts to control the mechanical properties of these bacteria, which are Vibrio cholera, into these porous mediums that we control the strength, the elastic modulus of them. We're also doing these biomineralized composites where we have these bacteria that produce calcium carbonate precipitates and looking at how the rheology changes with concentration of the bacteria. And then the last one is the self-healing composites, where the bacteria migrate to the air interfaces to form cellulose and looking at can we create self-healing porous composite systems. And so this is all the work that I've been doing up till now. And so really has given me these strengths in polymer mechanics, instrumentation development, and now really in biophysics and bioprinting. And so really if we go back to that bike picture that I talked about yesterday, which I think is still one of my favorite examples, and I know a lot of people do biking, as we were just talking about before this, me talking about this right here. So we go back to that bike helmet example where we think about this as a composite material. And oftentimes if you hit your head, you have these micro defects and really these limitations of many of the current polymer systems where they're single use, or we have to throw that helmet away and they come from non-renewable resources. So a lot of these are petroleum-based products and we want to try to step away from that and look for a more sustainable resource. So really these next generation of materials, we can think about these polymer helmets being self-healing or self-sustaining, and that we can control their properties. And really, while we've been trying to do this as a grand challenge in the synthetic systems, there's been a new focus more on biological systems because they already have this toolkit specifically on that field of engineered living materials. Where for this, we thinking about using biological systems and leveraging them to create functional materials in different applications through genetic engineering, where we have these cells that we can control their global mechanical properties. But most of the work that's been done is being just tuning one thing in the genetics and looking how it changes those properties. And because these are such complex systems where they respond to mechanical response, to environmental stimuli, they can control and change what their mechanical properties are dynamically. So they've been very difficult to understand how the molecular changes control the mechanical properties. So really in this independent process, I'm really thinking about characterizing the mechanical properties of these biofilms, and then leveraging those systems to create functional living polymeric materials for different applications. And really this vision is around this lab, which is the Huli Materials Lab, where Huli means to change or to study, where Huli is this part of the taro plant or the stalk of it, where I mentioned yesterday where it's planted. So this is a very, it's used for poi. So I'm sure some of you've heard of that purple sticky stuff that is a very staple in Hawaii. And they also use the leaves for lau. So they'll cut off the leaves and plant the root and use those to make food, and then we'll plant the huli back and they'll create this regenerating cycle. So really my group lies a little bit more in detail than I talked yesterday on these two sides of one, the fundamental, looking at the mechanics of living materials, and then more on the fabrication side. So on the mechanics side, we're going to look at creating instrumentation through custom-built instrumentation to look at these mechanical properties, where we'll answer questions like how do the mechanical properties change and how and they relate to the composition, where you can think of these biofilms as bacteria and polysaccharides, which are the polymer system, but they also contain exo-DNA and these interesting protein composite complexes that can either have interesting bond behavior or assemble as proteins to become long polymer chains. So really thinking about these complex systems. So this is one area that we can further explore. If we can think also about different bacteria with different compositions in our skin, that we have more than one bacteria. There's probably 1,000 types of bacteria that integrate to create our biofilm systems. So then we're also going to probe in this area, like how do the mechanical properties change in response to the environment, as they're very dynamic in nature, where they can, one, they're very antibiotic resistant. So can we see how the mechanical properties change with antibiotics, and to one, remove bacteria, or look at how the concentration of liquid, because these are hydrogels most of the time, if we change the humidity in the system or the temperature or any other environmental stresses.
Kanani, can I ask a question?
Yeah. No, go ahead.
You can defer this to later if you're going to give us more detail in subsequent slides. But I was just curious, on a previous slide you had mentioned, and though I'm not deeply knowledgeable in this field at all, I could imagine that a lot of the work has been, let's tweak something and just see what happens. But it's hard to work out causal relationships or design paradigms because of the complexity. And I was just curious if you had, specific thoughts about how to break that current paradigm, and is it a question of screening huge libraries and doing combinatorial studies, or is it more of a detailed mechanistic study, and how you would go about that?
Yeah, that's a good question. I'm going to talk a little bit about it, but I can answer it now. So for the initial mechanics, we're going to do just one bacterial system where we're going to do gene knockout. So we can knock out the specific polymer substances that they produce. So we can have them only produce this polysaccharide without the cross-linking proteins in the system, or we can have them produce only the cross-linking proteins and then subsequently do these steps. And this is really inspired by Howard Stone and Bonnie Blazer's group, where they've done this and done the rheology and capillary peeling, but they haven't really gone into these mechanical tensile testing setups. So like that's kind of where the initial goal is, and then eventually potentially doing screening of libraries and maybe doing some machine learning to see where we need to look in these very complex systems.
Great, thanks.
Yeah, any other questions right now? I'm happy to take any. Anyone is welcome to jump in at any time.
Quick question about the machine learning. How would you go about machine learning?
Right now, I would probably collaborate with somebody. I would probably measure the properties. We know a little bit about the structure of what's in the biofilms, but they don't really know how they interact and interplay. So I think that's something that we have to think about, and how to get those data sets to put them into the model to pull it out. Because if we want to think about, if we only knock this one thing out, if we know how each of them work separately, which ones do we want to interplay? So I think that's something that we have to consider, because they each have a different mechanism. But right now that's like in the future, like 5+ years, or talking to more people who do machine learning, because I don't do it right now. but I think very interesting opportunities in these spaces.
Sounds good. Thank you.
Yeah. So for the other side of my group is more thinking about applications on fabrication. And we're going to look at one, doing programming of the properties either through the molecular scale, through genetic engineering. So we can think about doing synthetic biology, or we're taking and making these protein complexes where we have an amyloid and an interesting protein behavior that we functionalize them, or to do macroscopic patterning where we can do 3D bioprinting to control the spatial morphologies, or eventually potentially going into optogenetics, which has been now slowly being shown further as you can control the properties of specific bacteria by shining certain wavelengths of light on them. And so really this side of the group is to look at more on achieving these functional systems through these sustainable materials, thinking about potential healthcare applications, to soft robotics with actuators, if we can think about making these responsive to stimuli, and then potentially in the far distant future, the protective equipment. So really the first area of my group will be thinking about these extreme mechanics of biofilms. So biofilms are really used already in different applications from bioprocesses. So they're used in wastewater treatment. So this is more of a beneficial thought to filter out a lot of the antibiotics and clean our water systems. But there's also issues with biofilms, and I think that's where most people think about them, is they cause these infections and they are really antibiotic resistant. If you can think about this pacemaker, they have to replace them or clean them. And so can we think about how to look at what mechanical disturbances are necessary to remove these films? And so one of the things that when I first started in working with biofilms, I thought, oh, these are just goop. And I think this is almost the common knowledge. They're actually very hierarchical structure, complex systems. So we have this nice review where I pulled one of their nice schematics out here, where you can see that depending on where they're located compared to the surface, they have different components. And so one of the interesting things that has really been looked at in terms of the mechanical properties is they really only look either at the bulk or the local properties, where one of the common methods is rheology, where they take these bacteria from a plate and they group them all together. So you're losing that complex structure now. And now they're just measuring the bulk system. So they're not, and this is really what Howard Stone's group does. So they're not able to capture how this hierarchical structure impacts those mechanics in those systems. And really, the other way that rheometry is used, they also grow them onto the rheometer, and that takes a little while, and then they measure those properties. But the issue with that is even the biofilms are dependent on what substrate they grow. So they can change their mechanics just dependent on the stiffness of the substrate. So that's an interesting thing to probe. But the other one is, if you can probe local, is doing either microcantilever testing or AFM, where they do compression, or local tensile measurements, but they're only looking at that top layer and those properties, so they can't access that hierarchical structure again. So really these gaps and open questions is, one, what is this cohesive strength and how does it relate to these complex structures and these hierarchical structures? And then can we look at further understanding how they respond to the environment? If you think about these plates controlling the properties, where they've shown that osmotic pressure of these plates matter. So really, the first area of my group is going to look at, one, investigating the role of the matrix composition through gene knockout, and then looking at the hierarchical structures in the effects of the external environment on these tensile and fracture properties. And so really to do this, we're going to use the tensile tester that I developed in my PhD. So Tut here, where we have that cantilever-based system, but now we have an opportunity to, one, change out this dog bone shaped film to a biofilm and put on a liquid supported bath that we can control what the environment is. Either it can be a liquid growth media, or we can introduce contaminants in the system. And the nice thing about this instrument is that it's mostly off the shelf, where we can buy this linear actuator, this laser source, and the camera, and then everything else, I have the CAD files to 3D print or even machine them, which we've done with these baths before. And so this could be set up pretty fast on an optical table within 3 months.
Do you expect to have any issues with delamination? I think you said you used mica previously. Do
Yeah.
you expect that that would still work here?
Yeah, so I wouldn't use mica. So some of the Howard Stone's groups shown that they can take, they can grow their agar, they can grow their films, and they were doing adhesion measurements, but they can do capillary peeling. So if they just lift them off the water, so they take the film, they bring it into the water contact, and then they have a film on top of the liquid substrate. And so they've been able to show that you lose some of the adhesion protein. So that's an interesting thing. If we can measure potentially in the future, can we measure both the adhesion between that substrate and the film and the cohesion in that other system? And can we potentially separate those two properties in terms of that complex hierarchy?
Thank you.
Yeah. And so really one of the first steps here is we're going to quantify the relationship between the compositions and the mechanical properties. And we're going to use these Vibrio cholera mutants that I'm already comfortable with working with. So they're actually biosafety level 2. So that would be one thing I have to think about in terms of my lab. But this the reason why we would do these is because one, they're well quantified in terms of the rheology already, in terms of how the gene knockout. So it's just a nice initial protocol to check that we are getting similar responses to the rheology, and is it's are we seeing different responses in terms of the cohesion? And then looking at, because these are viscoelastic in nature, can we look at the strain rate effects and potentially put these into a dynamic mechanical analyzer and do DMA and see if we can see these viscoelastic effects. And then looking more in the terms of the external environment, for these initial ones, we would introduce antibiotics into the liquid growth media at different concentrations and see if we can measure a difference response in terms of the mechanical properties. But there's a lot of opportunities in the future to go further into different directions and look at different bacteria and look at different colonies. And so I'm very excited about playing in this space.
Kanani, I had a quick question about the mutants. So is the sort of response to the mutations like linear in terms of the material properties? And by that I mean, like, when you make the mutation, you know, it might be a gene that encodes for a particular structural element of, you know, the biofilm, but in deleting it, the cell might adjust by making more of other components. And so the question is, like, how do you deal with that sort of nonlinear effect on the composition of the knockouts?
That's
if it exists.
Yeah, I'm not sure if it exists. That's a very interesting question. I know they don't get a nonlinear response because they've shown, at least with the cells only, that it's very weak. And then if you have just the polysaccharide and none of the cross-linking proteins, they're weak, but they're very elastic, where they can be very, I guess we'll use the word tough, even though that's very subjective. But they get stronger if you have the cross-linking system. So I don't know. That's a very interesting question to think about, and I haven't quite thought about it, but I'll keep. Thanks, Jerome.
Cool.
Any other questions? Okay, so
I have one more. I was just
no, go ahead.
Curious, sorry.
No.
Because I was thinking about the biofilm, like the wastewater treatment. And so one question I had, like, biofilms are really cool because of the chemistries they can do. And a lot of the hierarchical structure sort of stabilizes them to environmental stress, you know, they keep churning out, they keep performing the chemistry. So one question I had is, like, to what extent is, like, the material property of the biofilm important, or is it just the chemistry? And then, like, the related question, which I think maybe is what you're talking about, is, like, does the surface on which they grow affect the chemistry that they do, or, like, the robustness of the film? So is it, like, the material properties of the surface on which they grow that affects the chemistry, which is the cool part, or are there material properties of the biofilm itself that that are important? This might be a really naive question. So
no, so I'll answer the first one in terms of if we think about the wastewater treatment. So yeah, the chemistry is very important, but a lot of times they have to go and replace the films. because they encounter the same mechanical stresses. So they encounter shear, tensile, and compression in those flow cells, but they really only, most of the work's been focused more on the compression and the shear work. And so I think understanding the tensile is important, so if we can make mechanically robust films to have them continue to do that chemistry. So I think one, the mechanics are important. I think they're just often forgotten about, or well, also they're difficult to handle and measure, and that's part of the reason why it hasn't been done. And most of the work before in bacteria was mostly on the non-bioforming bacteria, and it's really been growing in terms of the biofilm sector of the area of the biology. And then for the second one about the surface and it's growing, at least for Vibrio, it's mostly dependent on the osmotic pressure. So how much of an, trying to think, it's essentially how much nutrient in liquid, if I remember properly, that the biofilm has access to that impacts those properties. So if you have, and the Stone Group has done some really nice work in terms of looking this and with wrinkling, where they've shown that I think the elastic modulus increases with the stiffness of the matrix. 1 second, I'm gonna lose my computer if I don't tell it not to restart. Does that answer your question exactly?
Yeah.
I'm not sure about the chemistry of it as much, but that's a good question. I think they do react they do respond to and change their chemistry and mechanical response, but I'm not sure what they do. So this has potential to be funded by NSF CBET. And then potentially, if we think about the antibiotics and the treatment of the NIH, NIGMS, and then for Boulder, we can think about the instrumentation shop to create these machine parts, and then co-sync of creating these cantilevers are actually cover glass that we have evaporated aluminum. So we need at least the nanofab to create these cantilever systems, and then almost lies under the umbrella if we think about the medical applications in the BioFrontiers Institute. And it has potential not only to collaborate with these people, if we can think about wastewater treatment and biofilm mechanics space, and Anushree Chatterjee on quantum dot antibiotics, but I probably will have this as open instrumentation to look at other things other than biofilms, and really add to that collaboration that I've heard much about in the last 2 days. So with that, this is the first project. If anyone has any more questions, unless I can move to the next one. Okay, so the next area is more on looking at the functional materials, on programming the mechanical properties of these biohybrid hydrogels, and really was inspired by this work for thinking about these cell-to-cell connections in mostly mammalian cells. So I'm going to be looking these into biological systems, where the connection of a mammalian cell to a ECM, or that extracellular matrix, is often done through these catch bond behaviors, and the cell-to-cell adhesion is through these catch bonds. And where catch bonds are different than what we think of a typical slip bond, which is what is defined as a covalent bond in biology, where when you pull it, it gets weaker. But with catch bonds, they have 2 stress states. And so you can kind of think about this as a finger trap. So those toy finger traps, when you stretch them, it gets harder to remove your fingers, but when you release them, it's easier to remove. So we can think, and really it's been not widely achieved in terms of the synthetically system, because these are complex protein systems where they bind to either a sugar or to the actin in the system, and they are able to pull and change their conformation depending on the force applied. And I think one of the reasons I'm also really inspired by this is there's some interesting work by Ana Spalaza's group and Sinan Ketan on the simulation sides, where they've integrated these cells, these catch bonds into nanoparticle matrices, where here we have a force versus strain, where there are 3 different strain rates. So if you increase your strain rate, we typically think of a system or a polymer as you pull it and you pull it faster and faster, they get more brittle. But in these systems, as you pull it faster at higher strain rates, they get more tough. So thinking about this in terms of protective equipment potentially. So really, it's been difficult to create these top-down techniques using polymeric materials, though there's been some work to achieve these, but right now, a lot of the molecules get created in the single, just as small molecules, and they do, when they looked at them under, sonication, where they stretched them, they only broke at the linker between the chain and that small molecule that should give you these catch-on bond behaviors. So why just try create these synthetically? We're going to try and use biology to create these catch bonds since they already exist in these systems. So really, can we make these tunable polymeric hydrogel systems by integrating these biological catch bonds? So really going to do this through engineering these catch bond networks through this system called the biofilm integrated nanofiber display system, which is a E. coli where we will have a plasmid vector of this complex, the FIMH complex, where FIMH is a catch bond adhesion protein that's used by E. coli to actually cause urinary tract infections. So it's been shown that under shear, it increases in strength, and as part of the reason why they're so infectious. And so we're gonna use these catch bonds, these FIMH behaviors, and then attach them and functionalize these amyloid proteins that can self-assemble into protein polymer-like strands, and then have these mannose PEG, which is MPEG or polyethylene glycol, if we can control the architecture, where the end linkers are these mannose, which is what FIMH binds to. So if we, think about, can we characterize these mechanical properties, which we'll do through rheology, potentially tensile testing and DMA, where we can get interesting rate effects. And we can, change the cross-linking percentage in our bath, where we'll take these and grow them in that filter in this liquid culture, and then we can filter them out and create these bacterial hydrogels. And then change the percentage of these FIMH or these cross catch bonds in these systems, or where we can do this by co-culturing one without those and one with those. So it'd be a random copolymer essentially. And then we can look at these in terms of live or cell-free, where we can wash out the cells in these systems and then look at these mechanics. Any questions?
Yeah, I have a question. So in the catch bond, is your definition that it has to get stronger? I mean, so in your cartoon, you had the second peak be higher than the first one. Yeah, with the straight. I'm sorry, that's lifetime.
Yeah,
But,
that's a bond lifetime.
Yeah, does it have to be stronger or longer lifetime, or are you okay with kind of a more extended profile? I guess I was thinking that was stress strain, but yeah.
Yeah, this isn't stress. This is think about these 2 lifetimes, but yeah. I think that's an interesting thought. I think right now with my definition, it's that they get stronger on that second lifetime response. And that's kind of where the catch bond behavior is, but it could potentially, you can think about it in terms of dynamic bonds, which have been growing as the field, where they're able to rejoin after, like you can think about those meta-coordinated cross-linkers where they get stronger, but more looking at, can we get these stronger second lifetime bonds?
Yeah, I mean, look, I mean, it's an interesting idea, but one thing that worries me about this design is you're kind of, I mean, it sounds like this is an example of a catch bond, but if you're defining yourself too narrowly, you may be missing out on a lot of material space.
Yeah.
I mean, when you say catch bond, on the chemistry side, I immediately go to things like, repeating peptides or Titan mimics, you know, like the Given Guan, like the Niels Holm Anderson with the metal complexes, like you were saying, where the toughness, you have basically the same strength on a per bond basis, but it's just repeating. And then as you stretch it, you get the entropic dissipation, which is where the energy dissipation comes in. But if you're defining that the bond itself is going to get stronger on a per bond basis, that's much harder to do. So I mean, kind of I ask you, like, what's your ultimate goal? Or do you have an ultimate goal? Or are you trying to explore the space? Or
I'm mostly trying to explore the space right now. I think it's just highly the network in terms of putting these type of catch bonds into networks hasn't really been explored. Mostly they've been the catch bonds have looked on the single molecule level and not putting them experimentally into these systems. And will we see the same behavior as they see in the simulations? But yeah, I agree. There's a bunch of work like Niels Holm Anderson in terms of the other types of bonds too. But I was just curious, can we look at these systems and will we see a different behavior than we classically see?
And I guess as a related question, and then I'll let you get back to it. How would you, I mean, so one of the problems you see with biofilms, I mean, it's hard enough to make a hydrogel work the same way every single time. And biofilms just take the chaos to another level. I mean, there is ordering inside, but let's say batch to batch variability with a first year graduate student in realistic case. How would you even know if you had a catch bond? Or is it just you have a material that's really good at energy dissipation? Because I can't imagine you would ever get a sawtooth pattern in your stress strain curve with something this disordered.
Yeah, and I think that's a good question in terms of if you have a catch bond. I think one of the ways we could step it back in terms of checking is take just nanoparticles that are coated with the mannose and introduce and just have the E. coli produce those FIMH proteins and then look, do we see that same behavior where now we've just taken the biofilm out of it and we're just producing that protein complex.
I mean, are you comfortable with things like force extension microscopy that you could use to do this on a single or few polymer level and then try to multiply that into the biofilm?
Yeah, I think that I don't have as much expertise in that, but I think that we could definitely explore that.
Okay. All right. Thank you.
Thank you. Okay, so with that, this looking at applying this for NSF DMR, potentially for the 3M non-tenure faculty award, and then if it's these interesting behaviors, looking through doing the HSSP assistant faculty grant where you get people internationally to come collaborate with you. And then this kind of falls under the soft materials research and has many interesting potential collaborations if we can think about in other biomolecular crosslinkers with Jerome or with Joel in terms of protein and polymer conjugates, or if we can do single molecule fluorescence and seeing that's a potential way of that we can probe these catch bonds are occurring in the systems, and then potentially looking at these in terms of tissue engineering is oftentimes skin fragilities, sometimes related to these proteins misfolding these systems. Any other questions in this area? If not, I'll go to the next one. So the last initial area of my group would be looking at these stimuli responsive living materials, where we can think about these bacteria, as I said, they respond to different stimuli and they'll change their composite systems, where this has really been a growing and emerging field, more looking at the stimuli responsive. I think a paper just came out in Nature Materials the other day where they were looking at cellulose, where you can introduce low concentrations of stimuli, either pollutants or biochemicals, and either create these biosensors or have these as drug deliveries and potentially soft robotics. And so thinking about these, most of the work, though there's now been a little bit more has been done on non-biofilm forming bacteria. So they put these bacteria into these polymeric, essentially mimics of the polymeric substance. So they take synthetic systems and they embed the bacteria and use them as those biosensors. And so really this is due to the fact that one, it's difficult to 3D print, spatially program these biofilms into systems, which is the work I'm working on right now at, in the data lab, is can we create these spatial morphologies and create these mechanical rules so that we can control those. And so really this goal of this area is, can we make these program spatial morphology in the macroscopic to create these protein complex shape changes within these polymer composite systems. And so this area is really looking at, one, can we quantify this relationship between this composition, again, just doing gene knockouts, and stimuli responsive shape, where we can do this with just E. coli K12, which is the basic E. coli that is used. So it's a biosafety level 1. And then can we introduce different stimuli, either temperature or pH, where we're having these films printed onto either PDMS substrate or some other substrate, and see if we can get differential swelling and what responses are we going to have. Are there any questions so far? Just making sure. And then really look at, now that we know how each of these mutants respond to the properties, can we print multiple different strains of the same bacteria, but now with just different polymer substance with different stimuli response into different complex shape behaviors. So if we have the same bacteria, just different strains, can we get interesting bending and behavior? And really the eventual goal is, can we create these more into complex 3D architectures, where these are mostly, we think of biofilms on 2D planar. Can we eventually make some interesting structures that are self-regulating in these systems?
Kanani, could I break in a fairly high level question here? So in the first project, I think I got very clearly that your expertise in micromechanical characterization kind of brought a new angle on this biofilm. And I was just wondering if you could make kind of a similar case here, as far as, I don't know the field that well, but I know a few people like Neil Joshi and others who are working on trying to engineer biofilms as materials. And so kind of what the distinguishing characteristics and unique angles are. Yeah.
So I think if we compare to Neil Joshi's group, which has great work and inspired a lot of my thoughts on these things, is that they really are still struggling. And I've talked with Neil about this in terms of the structure property relationships, because they're such complex systems. And so really trying to bring my polymer physics and polymer engineering knowledge and how the different behaviors, if we think about these ultra thin systems, relate to the mechanics that we're seeing, because I've thought about these complex changes before in a different sense.
Okay, thanks.
Yeah. Any other questions?
Hey, can I ask a quick question, Kanani?
Oh, yeah.
So I feel like one thing maybe I haven't heard you talk so much about, and I apologize if I missed it, is sort of like the kinetics of these types of, the timescales at which, kind of the different observations and phenomena are actually happening. Right? And so, I'm just curious, I guess, these bacteria obviously take time to respond to things and then produce chemicals that might then lead to the types of changes that you're hoping to see or you're proposing here. And I'm just interested kind of in general, what sort of timescales are reasonable, and is this a potential limiting factor for some of the kind of applications maybe that you're thinking about?
That's a very good question. In terms of the kinetics, I'm not 100% sure, so I'll give a stab at it, but I can't guarantee. So I think it's very dependent, one, on the strain and then what they're sensing. I think they can be on the order of minutes, but I'm not sure. I really am unsure about that. So,
Okay.
But that's a good question, and I should definitely look that up.
Yeah, it's more just, I'm just interested.
Yeah.
I mean, it just seems like it's an important parameter in thinking about
No,
using biofilms for some of the kind of, for some of the potential applications that you're
Yeah.
suggesting. And so, maybe it's just more of a suggestion that it might be something to put some thought into if you hadn't.
Yeah, I definitely agree. And I haven't put that much thought into it,
Yeah.
but I should.
No, it's totally fine. Yeah, thank you.
Thank you. Any others? If not, so with that, this will fall under the, again, NSF-DMR. The Air Force Research Lab actually has a call out for sensors and biological systems, using biological systems. So that falls under the DoD there. And then potentially looking at these under using the light microscopy core facility eventually, because we can look at if we have these fluorescently labeled and look at the interface, because sometimes with bacterial, they'll self-organize and self-assemble into interesting structures, and then potential collaborators. And if not, if there's any more questions, I'm going to go into the teaching and mentoring DEI slides.
Yeah, I just want to
yeah,
jump in real quick.
Mm-hmm.
So can you just give us in a 1 or 2 sentences what your, the overarching vision for your lab? So when you go up for tenure, what your lab is going to be known for, and what kind of is the big advance that your lab is going to make?
Mm-hmm. So I think it's really one, looking at biofilms and understanding these mechanical properties of these systems, and then using that knowledge from that mechanical properties to make this potentially understand how to make functional materials.
Okay, thank you.
Yeah.
I have a question, I guess,
yeah.
or just, I don't know if it's an idea, just from Joel's comment. So, yeah, like, I mean, if you think about like a developing embryo or like an organ, there's like mechanical properties that guide sort of the structuring. And so I was wondering if there's something you can do here where you're using mechanical stimuli on different time scales to guide the structuring of the biofilms. If there's anything neat in that, or, because if you kind of think about like what you could do to guide the biofilms in an unnatural way, it seems like mechanical structuring, you can create these large mechanical cues on different time scales, so you have a lot of control, and that may allow you to create, I don't know, different biofilms with different organizations.
Yeah, I think that's a very interesting. Yeah, I think that's a very interesting idea in terms of looking at that. If we can put them under different mechanical stress. That was one of the parts of my proposal here at Princeton, was seeing if I wrinkled them, could I get them to do interesting behaviors, and really more looking at the spatial organization. So I am definitely interested in that. I'm supposed to be working on that. We'll probably not get to it, so potentially in the future.
Yeah, I mean, like even just retention of shape, could be cool. That's an interesting outcome. Yeah, anyway.
Right. So Kanani, I think you said you were just about to move on, but just looking at the clock, I know some folks will have to leave right at 9:30. So
yeah.
Thanks.
Okay. So for teaching, so my teaching philosophy is really trying to use active learning tools. So thinking about think, pair, share, discussion groups, and project-based learning with some lectures involved to really have a student-centered focus, really motivating and trying to teach the students best. And this is, I've been working where I haven't had a much teaching experience, but I've sought out different programs. So this is the NSF Academe workshop that I did 2 years ago down at University of Mississippi. Yeah, University of Mississippi. I think, yeah, it was long 2 weeks, where we did a week-long teaching pedagogy workshop, and then we also did a week-long research intensive workshop with mostly underrepresented minorities. I also have my mentoring philosophy is really to empower students to, one, succeed in their own research goals and their own professional development goals. Are really looking at what they want to achieve and helping them achieve those goals, and really try to provide a supportive environment so that they have that independence to, one, work on projects that they're interested in, but also still work on the projects that I have funding for. And this is the grad student who took over my PhD work, Cindy, in the lab. So really the courses at in the CU Chemie department, be interested in teaching is one, the undergraduate fluid mechanics class. And then the materials class, which I actually looked at the syllabus online. I had to laugh because it's the same textbook that I learned for my bachelor's degree, like intro to materials engineering. So very familiar with that book, the Callister, and then transport phenomena for the graduate work. And then with my background in polymer science and engineering from my PhD, would be interested in teaching that polymer engineering course. And really for potential future courses is creating a course based on my research on engineered living materials, but more on the diversity and inclusion efforts, is trying to create a module or elective, both at the graduate and undergraduate level, potentially a combination course on designing K-12 outreach to look at, one, how do we communicate our research to different levels, and then thinking about different medias and not just in front of the classroom, especially as we now think about virtual environments where we're on Zoom, through Twitter, through creating Wikipedia articles, and really teaching on these different communication skills. It's become more and more important to have the science communication with the general public. And so really, because this is a diversity and inclusion search, you have to definitely highlight some of the work I did during my graduate work, my undergrad and my graduate work, where I was a part of the Society of Women Engineers, or SWE here, and where I did outreach through all 4 years of my time at RPI. And then was a part of the UMass PSC outreach program, where I volunteered to teach about polymer in science. And then now I've been really working with, on the more of a national level, with the American Science and Engineering Society at their national conference, where I presented last fall about what materials engineering is to get more natives interested in STEM. And then in this last year, I started becoming a wiki editor, and this is, you have an opportunity to edit. Anybody can go and learn to become a wiki editor. And part of the reason that I've become one is that only 17% of the biographies on Wikipedia are of women. So it's very underrepresentative in terms of the women and in terms of these biographies. And really one of the ones that I got to make this year was for the late Anne Mays, who was the first tenured professor in the materials department at MIT. And Monica de la Cruz graciously gave me this photo of her to put on the website. So it's been very fun looking at these potential opportunities for outreach, and then looking further in the future is continuing to do the course, but do outreach with the Boulder, Denver area and the Engineer Me, and then working with the Bold Center and potentially continue to work on with ACES. So really, as I am part Native Hawaiian, I'm very passionate about natives in STEM. And if we look at the Colorado Indigenous population, it's a 1.8%. But if you look at the undergrad makeup and the grad makeup, we can see that we're not at that representative scale of relating how many people. And so really going out to try, one, to the tribal colleges over in the neighboring states, or trying to create that network with the other, with those native indigenous tribes in the area, and working with the Colorado Diversity Initiative to go to ACES national conference, where Colorado is already a top 200 school within their society. We just don't attend, and there's already a local chapter within the school with the students. And then one of the things that has been emerging recently is the Society of Indigenous Physicists that I've become a part of. And there's about 40 Indigenous physicists, from Native Hawaiians to Native Americans across the country, who physicists is broadly defined from engineering to physics to many different fields. And so with that, I'm also very passionate about that mentoring more, of where I've mentored undergraduate students, 3 in the lab, but also 2 externally through the program at UMass Amherst, which was the retention program, which was originally called the Northeast Alliance for Graduate Alliance and Graduate Acceleration Professoriate, I think. Never remember what NEGAP stands for.
Graduate
Brian,
education and the professoriate, yeah.
And professoriate. That's what happens when you name things and then you just go by your acronym. And then that's become the Spalding Smith program. So I got the opportunity to mentor 2 of the students and the PSC department, who were, one was a Puerto Rican woman and the other one was a South Pacific Islander. So very excited to watch their trajectory, and they've been doing very well so far in the program. And I've watched the importance of that program to see. I have had colleagues who were thinking about dropping out of going to grad school, and that having that support circle and those mentors kept them in the graduate program. So that's very important to me. And I've also become a part of this Lighting the Pathways program at ACES, which is advancing the professoriate specifically for natives in STEM from the undergraduate level up to the professoriate level. So we have freshmen in college to postdocs and senior faculty to deans, where we have mentoring for once a month. And so I'll have opportunities to mentor either undergraduates or graduate students at, when I become a professor, but I already have a mentor right now who's a dean at a different university. And so it's been very exciting to see the opportunities there and meeting other natives in STEM. And that's one of the biggest pools of natives in STEM across the nation, where it's gotten funded 2 years from NSF for 5 years and another 5 years from NSF, because it's the largest pool and they can study the impacts of having these mentoring programs. So there's in the first cohort with the 5 years, they had 100 natives in STEM, which is one of the largest, if you think about often what we have typically in these pools, where they don't even report them in the NSF numbers. And so really in the future, looking at working with the STEM routes program, where they're to do the Uplift research program and funding undergraduate research, again, working the BOLD Center, and then with that summer multicultural access to research and training, the SMART program. And then really creating diverse and inclusive spaces. And this is more come out of the Black Lives Matter movement back in June, where I started working with the data group to have a diversity and inclusion subgroup that we run now once a month, but we were running it every other week at the beginning, and really having these conversations, where this led to a discussion group that we created called at the Virtual Polymer Physics Symposium that I ran this summer in replacement of the Gordon Research Conference that was supposed to happen. So these are all of our discussion leaders. You can see there's Alexa, who was one of our discussion leaders, and really providing these opportunities and spaces for people to have these discussions. And so we had 70 early career researchers be involved in that, and and then we took those conversations and what we made it into a document and submitted it to the executive committee at the polymer physics section in the American Physics Society. And then also that came out of it was creating a space for these early career researchers to have a polymer physics Slack, where we're able to communicate about jobs, just have a pipeline, especially since we're not seeing each other with COVID and conferences. And so I've seen the impact of that, where people have learned about grants that they hadn't learned, and awards that they hadn't applied to before, and receiving those. And so in the future, my group will continue to have a diversity and inclusion subgroup. And then during group meetings, they'll have this 1-minute diversity slide that was created out of Rachel Siegelman's group and the Molecular Foundry, where they present something, either about themselves or something they're excited about in terms of diversity, and then potentially looking at joining the DEI committee in the department. And with that, I welcome any questions, but that is it.
Great, thank you, Kanani. More questions? I really like the ACES, the plans and all the things you've been doing with ACES. I have to admit, I embarrassingly was not very aware of that organization or how active they were. So thanks for.
Yeah, I mostly became more active with them at the end of my PhD. I've been aware of them But the other one is SACNIS, the Society of, I always forget what that one stands for. So those are the two like indigenous societies. I'm just more active on the ACES side.
Society for the Advancement of Chicano, Hispanic, and Native American Scientists.
And that's a bigger organization, or this is a little bit smaller. But they have around, I think, so they also at their national conference, it's an opportunity to recruit undergraduates too, because they bring in high schoolers. So I think around like 200 high schoolers go to these conferences and graduate students. So it has those opportunities. And if your student presents a poster, they get free registration. So they really try to encourage students to go and network.
Just quickly, how have you found Slack as a networking tool for large groups? I mean, I only personally have experience with Slack with things like a research group or, you know, a group of maybe 6 people. Is it useful for, you know, groups of 50, 100?
Yeah,
or is
so we
it
have
just kind
over,
of like a LinkedIn, just like something that's kind of there? And
I think it, yeah, I think it's what you make of it, kind of like LinkedIn. We have over 200